So it's a great pleasure to be back here in Tirupati. Incidentally, when I was first admitted to fellow to the Indian Academy of Sciences, it was here in Tirupati. That was in 2002, I think. There was no IISER, there was no IIT, but it's great to be back. So what I'll do in the next 35 minutes is to tell you about some work in my lab that we have done over the years. And it's on enhancing the stability and efficacy of vaccines, primarily for respiratory viruses. So since this is a very general audience, the kind of work we do is pretty simple to understand. And so if you don't understand it, it's really my limitation, not yours. There are a few very simple principles that guide vaccine design. And the first of it has to do with the relationship between a protein sequence, its structure, and its stability. So proteins, as you probably all know, are biopolymers, and they are composed of 20 different naturally occurring amino acids. So these are the letters in the protein alphabet. And the protein sequence can be inferred through the genetic code from the gene sequence in a straightforward way. Many proteins, not all, in fact many don't, but many important proteins adopt a well-defined three-dimensional structure in which their atoms are precisely located in space. And of course, that's a time average structure. These macromolecular structures undergo dynamic fluctuations, and this structure is critical for the protein function. So a protein has to bind to other molecules, to other proteins, to small molecules, to nucleic acids. All of these are important for the way that proteins function, and their three-dimensional structure is important because typically when you have binding between two molecules, their shapes tend to be complementary. Now, in many cases, it's now possible to infer the structure from the sequence. And the great, this was an intractable problem for many years until with the convergence of enough structural data in the protein data bank and developments in artificial intelligence and machine learning led to a solution to the problem in the form of pattern recognition. And so now, for many proteins, you can, in fact, predict the structure. Another very important property of the protein is the free energy difference between the folded and the unfolded states of the protein. So as I said, proteins, many of them adopt well-defined three-dimensional structures, and even those that don't, once they bind to a binding partner, do adopt a well-defined structure. Now, that structure is always in equilibrium with an unfolded linear polypeptide chain. And the equilibrium for a folded protein is typically shifted to the side of the folded structure, but there is nevertheless an equilibrium. And this is very important because how long the protein lasts in the cell often has to do with the stability of the protein, which we can measure in the test tube. Not always, but many times. The stability of a globular protein can be quantitated in terms of the free energy of folding or the free energy of unfolding, which is related to the equilibrium constant in a straightforward way. And for monomeric proteins, this free energy of unfolding is a function of temperature. So as you heat up the protein, the equilibrium shifts to the more disordered state. And very interestingly, for many proteins, this equilibrium also shifts to the disordered state when you go to low temperature. And that's called cold denaturation, and this is typically below 0 degrees centigrade, so we don't observe it. And so you get this so-called stability curve. Now, what does all this have to do with vaccines? So a vaccine typically elicits antibodies, and neutralizing antibodies are, when I'm talking in the context of a virus, a neutralizing antibody is one that's directed against the major viral surface protein, and it binds to the surface protein and prevents the virus from entering cells. And this is particularly true of many important viruses which are surrounded by a lipid coat. So in order for the virus to enter the cell, the two membranes, the membrane on the virus and the membrane on the cell have to fuse to each other so that the genetic material in the virus can go inside the cell. Now, this process of the fusion of two negatively charged lipid membranes is energetically unfavorable because you have two negatively charged species which have to come close to each other, and there is obviously electrostatic repulsion between the two. And so in order to overcome this unfavorable energetic barrier, there has to be a driving force. And the driving force typically for many viral proteins happens in the form of a shape change of the protein. So most normal proteins are in a single folded state, but viral surface proteins, which are important for viral infection, are what are called metastable. That is, the state that is found in the natural context on the virus is not the most stable state. And so when you make the protein in the laboratory in the test tube, very often these proteins, instead of forming the desired so-called pre-fusion state, go into the post-fusion state. And so you need to figure out ways to stabilize these metastable proteins so that they are in their native conformation. And so I'll mostly be talking about vaccines that elicit antibodies, but of course there's another very important arm of the immune system, which are T cells, and they also contribute to protection, but I won't talk about them at all. Okay. So I'll just illustrate some of these shape changes with a virus that we work with called respiratory syncytial virus. So the important protein on the surface of the virus is this so-called F protein, which is in blue. And this is the shape of the F protein as it is on the surface of the virus, and it's in a relatively compact shape. Now, in order for the fusion of viral and cellular membranes to occur, it transitions to this elongated state, and that process releases a lot of free energy, which compensates for the unfavorable lipid mixing. Now, why is this important for a vaccine? So it turns out, of course, that the antibodies that you want are ones that bind to this state before the infection has actually occurred. And you don't want, in fact, antibodies that bind to this other state, because at best they are useless, and at worst, what can happen is these kind of antibodies can actually help infection. And that happens through mechanisms which I don't really have time to explain right now. But that's why it's critical that when you make a vaccine, the protein should be in this conformation. And this is just not just a theoretical thing. So for example, the RSV vaccine, the most common way that vaccines used to be made earlier and continue to be made is by inactivating the virus. And when you inactivate the virus, in some cases, if there's a lot of this incorrect conformation around, you will get antibodies that bind to that conformation when it's used as a vaccine. So when the RSV vaccine was made in the 1960s by inactivating the virus, and it was given to children, some of the children died because the vaccine, instead of preventing infection, actually enhanced infection. And the consequence of all this was that viral vaccine development for RSV, which kills over 120,000 children each year, most of them in low and middle income countries, was stalled for about 60 years until people figured out how to deal with this issue. So the RSV vaccine only recently became available in 2023. They are priced at over $200 a dose, and so they are obviously unaffordable for the people who need it most. And so this is something, among other things, that we are trying to address. Okay. So now, some of the things I'll discuss in my talk, you all know about SARS-CoV-2, and I'll use it only as an example of other things that we do. So, although in India nobody's vaccinated for COVID anymore, but really there is an ongoing requirement for periodic boosters for the very young, for the elderly and those with comorbidities, which is a large number of people. Now, worldwide, the mRNA vaccines didn't reach the vast majority of the world's population because those formulations required frozen storage. And so the worldwide vaccine coverage was highly non. It turned out not to matter so much, because in places where there was very poor vaccine coverage, it tended to happen in countries with a very, very young average age. And so, they were not as badly affected as they might have been. So, despite all the transformative advances in AI and machine learning, some of which you'll hear about in the next talk, and the ability to predict protein structures, the prediction of stabilizing mutations remains difficult, simply because there's not quite enough data on this, and it's a problem to be solved. And so there is a need for safe and inexpensive vaccines, both for SARS-CoV-2 as well as other sarbecoviruses and other respiratory viruses that can be administered to most of the world's population. Okay. So the tool that we help to develop and we use frequently in our work is something called deep mutational scanning. So the idea is that you have a protein of interest, and now you don't know which changes to make in the protein so that it becomes more stable. So what we can instead do is to create a library of all possible single point mutants of the protein and screen it. And the other advance that has helped over the past decade is the development of ability to simultaneously get the DNA sequence of a large number of different DNA molecules in a mixture. So this has been really transformative. Unfortunately, not yet awarded with a Nobel Prize, but it's long overdue. And this ability allows us to screen the libraries before and after they are put through some kind of screen. And by so doing, by counting which mutants pass the screen, you are able to assess the contribution of every mutant in the library to whatever your property of interest is. That can be the activity of an enzyme, it can be the ability of the protein to bind to a partner, and it has not been easy to convert that into a stability readout, but we have been able to do that. I won't again explain how. And so we were one of the earliest to, not the first, but one of the very, very early people to use this methodology, and actually the first to use it for a complete protein. And our interest was, this was well before AlphaFold, was to see if we knew the effect of every single point mutation in a protein, could we predict the structure? And so we've done a bunch of work over the years on this issue. But now with things like AlphaFold, some of that work is irrelevant. But the readout from this kind of an experiment is, so these are the residue positions in the protein. So let's say the protein has 100 different positions, and at each position we can put in one of 20 different amino acids, and we can score the effect of the mutation on whatever biological property we happen to measure in the screen. And so when you do, we can get stability information for all these different point mutants, and therefore identify each of the point mutants that individually enhances the stability of the protein. And this is through a genetic screen, so then we can make those proteins that we predicted to be more stable, and we can actually measure their thermal transitions. So when proteins melt, you get a change in some property, and the midpoint of this is the melting temperature of the protein. So you can see that we were able to make, in black is the wild type unmutated protein, and by making different point mutations, we were able to increase the stability of the protein by quite a large amount, or almost 15 to 20 degrees, and we could combine different point mutants and get large increases in the protein stability. So this is a way of enhancing protein stability in a high throughput way. Now, during the pandemic, you're all familiar with the SARS-CoV-2 virus. This is the spike protein of the virus, which is the target of all vaccines. And in particular, this is a region in red called the receptor binding domain. And the receptor binding domain binds to a molecule called ACE2 on the surface of our cells, and that triggers the process of viral entry. So we reasoned very early on in the pandemic that this would be the best target for a vaccine, and in fact that later turned out to be true. And so we made different variants of this protein. And what we found, very interestingly, was that when we freeze-dried the protein, you could heat it to 100 degrees centigrade and cool it down, and it still regained its original shape and activity. But the molecule by itself wasn't very good at making antibodies when used in a vaccine. So we attempted to enhance the immunogenicity by introducing stabilizing mutations into the receptor binding domain. Okay? So through the methods that I described. So now these are again thermal transitions of the wild type in red and the mutated one, and you get about an 8 or 9 degree shift in the thermal stability. But what's very interesting is, so this is it's a little bit technical. This is on a log scale. This is the dilution of sera from an immunized animal when you immunize with a molecule that you need to inhibit viral infection in cell culture. So you can see that with the stabilized mutant, the sera are 100-fold more potent. So even though we increase the stability by just a relatively small amount, that leads to a very great enhancement in the neutralizing capacity of the sera, which translates into enhanced efficacy. And also, although the protein that we used was derived from the wild type, the so-called Wuhan strain of the virus, even for much later strains which came 2 years later, the sera could still neutralize those strains. Whereas when we took the wild type or unmutated receptor binding domain, the immunogenicity was lower, and we completely lost the ability to neutralize later strains of the virus. So this just summarizes what I said. So it shows that enhancing the stability of the protein, besides enhancing its properties in the test tube in terms of storage and other things, also enhances a biological property, namely the ability of the protein to elicit neutralizing antibodies in an animal model. Next, we wanted to see that if we actually stored the protein at 37 degrees, a slightly modified version of the protein, at 37 degrees, and then used it as a vaccine, so we stored it at 37 degrees for a month and used it as a vaccine, would it still be just as good? And it turned out that it was. So this is just, again, don't bother too much about, these are different viruses. This is the original strain of the virus. These are variants. Neutralization of a very diverse range of viruses, though with varying titers and independent of concentration. And when we compare the lyophilized and the non-lyophilized material, if you just look at the relative neutralizing titers, they don't change very much. So in other words, we could make this formulation, store it at 37 degrees for a month, and it was still quite completely efficacious. Not only that, it protected animals against challenge. So if we challenge mice who are not immunized, they rapidly lose weight and die. The vaccinated animals don't. And even if we do it with different strains of the virus, it doesn't matter at all. And this is which is the surface protein of the SARS-CoV-2 virus. We subsequently applied it to several different proteins. And in each case, here the wild type protein without mutation is in red, and the mutated protein is in black. And we're showing different forms of thermal transition. And you can see we are consistently able to enhance the thermal stability using this approach. We're also able to enhance the thermal tolerance. So the wild type proteins are at the top, the mutant proteins at the bottom. And the data show that if you incubate at these different temperatures for an hour, cool it down, and then look at the melting, you can see that the mutant ones, they retain their shape and undergo the same kind of melting transition. The wild type ones don't. And so these are for a number of different important viral surface proteins. Okay. Not only does the thermal stability increase, but many cases the yield, which is shown on the y-axis, of the wild type versus the mutant protein increases very substantially, and this is of course a critical factor when you actually want to manufacture a vaccine. You have to be able to make it in large quantities. So these are different spike proteins from a number of different subecoviruses. SARS-CoV-2 is just one example of many hundreds of subecoviruses. And in the early 2000s, there was SARS-CoV-1. Now we've had SARS-CoV-2. And as I said, there are many other subecoviruses. They all have potential pandemic threats. So it's a forward procedure whereby which if we have another SARS-induced epidemic or pandemic, we can very quickly make a stable vaccine. The other thing I will talk about is an approach that we use to target where the sera in an immunized human or in infected human, on which part of the protein surface do they bind. So many of you here would be familiar with measuring binding or identifying a binding region when you have two molecules that bind to each other. That can be in a straightforward way through X-ray crystallography or cryo-EM or nuclear magnetic resonance or a variety of ways. But when you immunize and a protein in a body, the antibodies are not produced at they don't bind to just one location on the protein surface, they bind to many different locations. And in different people, the kinds of antibodies that are produced may be different. As the virus mutates, the regions where the antibodies bind may be different. So these are all different kinds of variations that we need to be able to account for. And so measuring in a complex mixture where you have many, many different kinds of antibodies, where this so-called polyclonal mixture is binding on the protein surface is not a straightforward thing to do. And it's important to be able to do this because you want to know when you either upon infection or upon vaccination, where all are the proteins binding, so that if now there's a viral mutation, you can figure out that this viral mutation may lead to a loss of efficacy of a vaccine. So here we have approached this by making, again, mutational libraries where we cover the surface of the protein with charged amino acid substitutions. And so you can imagine often when two proteins or two molecules bind to each other, there is nonpolar surface which is buried. And if you replace that nonpolar amino acid with a highly charged one, that's going to disrupt the binding. So what we do is we make a library where we put these charges at all different positions on the surface of the protein, and we see which of these surface mutations have disrupted binding to sera, and we can do this by very rapidly by using the next generation sequencing technology that I talked about, and that allows us to characterize the regions on the protein that are targeted in actual human sera. So the idea here is that we express the protein of yeast, and then we come in with either a monoclonal or polyclonal antibody, and we detect the binding of that antibody to the protein that's displayed on the yeast surface. And when we have a mixture of different mutants, we can see through next generation sequencing which of the mutants are binding to the sera and which of the mutants are not binding to the sera. And there's a way of very quickly doing it by barcoding the library, which I won't really talk about. So we did this in collaboration with Professor Anandam Mondal's lab at IIT Kharagpur. He had access to sera that were collected from individuals during the COVID pandemic. And these were what are called breakthrough infection sera. So all these people had been vaccinated, but then despite that, they got an infection. And so we were able to see, so these are three sera that we looked at. I mean, we looked at a bunch of these sera, and through the methodology I described, we were able to identify which were the residues in the protein which were being targeted in these polyclonal sera. And we could map those residues onto the protein structure. And you can see that although these are, of course, different individuals, many of the locations on the protein surfaces that were targeted were identical. Now, what was very interesting is that we could see where which regions of the protein surface were being targeted by the sera, and that would allow us to infer, when we had a different virus, a different variant of the virus, we could see if those residues had changed, then probably those sera would not be able to neutralize the virus. Whereas if those residues hadn't changed because of viral mutation, then our prediction was that indeed those sera would be able to neutralize the virus. And so what we did was to take a virus that evolved about a year and a half later than or 2 years, maybe a year and a half later, after the sera were collected. And we found that in contrast to what had been seen in the US and other Western countries, that we found that these residues that were targeted were conserved in these later strains of the virus. And so the sera we had predicted would be able to neutralize the virus, and in fact they did. So the sera that were generated earlier in India, at least in this set of people that we looked at, had much broader neutralization than sera in the US, and that's probably because these people were, the average age would have been younger because we're a relatively young country in terms of median age, and also because unlike the US where there was a lot more of much less contact, here in India, especially in the lower economic strata, people don't have the luxury of staying at home. And so there was continuous exposure to the virus, and we believe that drove evolution, or it increased the breadth of the antibody response so that it was targeted to more conserved regions of the protein. So if you look at the regions where these sera were binding as inferred by our experiments, they were all binding to a relatively conserved region of the protein, which is situated away from where the receptor is binding, the so-called ACE2 binding site. And so unlike what had been seen in the West, we found that the sera, at least in this population of people, were targeting this relatively conserved region of the virus, and therefore they were still able to neutralize later strains of the virus, and that may have contributed to the general observation that nobody here is now vaccinated, and there's of course ongoing viral evolution, but by and large people are not falling particularly sick. Okay. So now I would like to turn to some work that we have been doing with influenza virus. So influenza virus is another important respiratory virus. Unlike SARS-CoV-2, which has a single piece of RNA in the genome, here the influenza is a multi-stranded, what's called a segmented virus. And the important protein on the surface of the influenza virus is something called hemagglutinin, which binds to a receptor, which in this case is a carbohydrate called sialic acid. Now, influenza has many different so-called clades of the virus. The ones which are, there are 18 or 19 different clades of the virus. The ones that are primarily infecting humans are H1 and H3 and influenza B. So the vaccines that we have are typically, though very few people take them in India, are typically trivalent or quadrivalent vaccines. They need to have one hemagglutinin from H3, one from H1, and one or two from flu B. And every year these vaccines have to be changed because the virus keeps evolving. Okay? So every year, roughly 300,000 to 600,000 people die of seasonal influenza. And this number can be much larger in a pandemic. So for example, in the 1918 pandemic, which happened, there were, the death estimates are of the order of 50 to 100 million people. And a non-trivial fraction, India had the largest number of deaths in that pandemic, which happened just after World War I. So these pandemics can be really deadly. Now, currently there's a lot of concern about an H5N1. So you remember, so H5, H refers to the hemagglutinin, N refers to the neuraminidase, which is another surface protein on the flu virus. And there's a lot of concern about a recent H5N1 flu strain, which has crossed over from avian species, which has traditionally been infecting. You would all have heard of bird flu, but it's now crossed over into mammals. And this happened in 2022, 2023. And as you can see, the numbers are pretty staggering, right? So it's caused over 500 million deaths in birds all over the world. And it's present in approximately 70 mammalian species. And so it's, fortunately, it hasn't really crossed over into humans in a sustained way. There's been no human-to-human transmission of this particular strain, but it illustrates the dangers and the constant threats which we have to face and which are increasing due to global warming, due to globalization, deforestation, and so on and so forth. So several years ago, we figured out, it turns out in the flu virus, there is this protein hemagglutinin, and this is the part which binds to the receptor, the salic acid receptor. This part is involved in the viral fusion, the fusion of the viral and cellular membranes, and this lower part is relatively well conserved. So we figured out how to make just this lower part by itself, and we showed that when it was given as a vaccine, you can see this is in blue, residues are more conserved, red residues are less conserved. You can see this is a more conserved region of viral surface protein. And we showed that when we used it as in a vaccine, we could protect from challenge from very diverse strains of the virus. So we've continued working on that and working towards it to make better flu vaccines. Now, in parallel, the way flu vaccines are made is by inoculating the virus into hen's eggs. And each egg, and then the virus is grown in these eggs and extracted, and then it's chemically inactivated. And that's the vaccine, and it's some additional treatment is given, and then that's the vaccine which is given to people. Now, this is a relatively cumbersome process, and one egg produces one human dose of the vaccine. So for 1 billion people, you need 1 billion eggs. So clearly, it's not a scalable situation. And it's even worse if there is a avian origin flu pandemic, because avian viruses are toxic to eggs. They kill the eggs. So we need to have better ways of making vaccines. mRNA is one, but that has temperature stability issues. So we made a recombinant version of hemagglutinin in insect cells and showed that when we gave a vaccine made of this, so shown in the triangles is the vaccine formulation, which is the recombinant molecule, and this is the conventional vaccine, and we gave it to mice and challenged them with a very old virus. So these vaccines were based on current day strains, and the virus that was used in the challenge studies was either from 1982 or 2008. But essentially, we showed that the recombinant vaccine protected much better than the commercial egg-grown vaccine. So we could see improved protection in mice against very heterologous means with challenge with a very different strain of the virus. And we made mutations in the hemagglutinin to increase the expression. And this was formulated with an adjuvant. So when you actually start going into the clinic, you have to worry. So adjuvants are molecules that you can add to the vaccine formulation to enhance its efficacy and to enhance its immunogenicity. And it turns out there are very few vaccine adjuvants that are approved for human use. You can count them on the fingers of one hand. And most of these are held by large companies, by large pharma, and so you need to license out from them to use it, and it's not straightforward to get those licenses. So fortunately, the Gates Foundation had engaged to make generic versions of some of these vaccine adjuvants, and we were able to use one of them, which is an oil in water emulsion-based adjuvant. And this has been used in conventional flu vaccines in millions of people in Europe, so we knew it had a good safety record. So recently we carried out a clinical trial in Australia with this vaccine formulation, and we showed that the vaccine was safe. We didn't have the numbers to look at efficacy. This was just a safety trial, a phase 1 trial, and it was completed, and the vaccine was found to be safe and well tolerated. And the follow-on clinical development will be done in India with generous support from the Indian Council of Medical Research. So now just to come back to the H5 influenza vaccine, as I said, this is a potential for concern, and it's infected a large number of dairy cattle herds. Interestingly, only in the US, for some reason, other dairy cattle herds in Europe and other places, we haven't found evidence that this virus has crossed over into the cattle. And so when it goes into the cattle, it goes into the milk. So people need to, pasteurize the milk, heat it up before they drink it. So we and others have been working on approaches to deal with this potential pandemic threat. As I said, fortunately, so far it has not. There's been no case of human-to-human transmission documented. The number of deaths in humans from this particular strain have been very low. But nevertheless, it's an illustration of how rapidly these pandemic strains can arise, especially in a segmented virus like influenza virus, which infects multiple hosts. So when you have a virus which infects multiple hosts, you can have two different viruses recombining in the same host. And most of the time, that doesn't lead to infectious virus, but sometimes you can get virus that crosses the species barrier. And with increased contact between humans and animals and all the threats, environmental threats that we continue to face, and with global warming, these kind of events are going to happen more frequently because we'll come into contact with viruses that we normally would not come in contact with. Okay, so I'll end up here so that we have plenty of time for questions. So just to summarize, we want to show that we've developed methodology to relatively rapidly stabilize proteins through mutation, and we validated this in a number of different, we have validated this in a number of different proteins. And in the case of the COVID virus, by stabilizing the mutation, we increased its protective efficacy, and we got a lot of press on this. Unfortunately, no significant amount of money and time to do anything, though we were the first in the country to generate the vaccine formulation. And I'll come to that in a couple of slides. So using this novel epitope mapping approach, we were able to show that sera from breakthrough infections in India target a novel conserved epitope, and these sera were broadly neutralizing. And a few years ago in 2017, I co-founded a startup called Minvax, and along with Minvax, we have been able to develop a flu vaccine formulation and complete a phase 1 clinical trial. And we will continue our clinical development in India. And if the results are good, which they should be because it's worked very well in animals, but until we actually do it in humans on a large scale, we wouldn't know. This would be the first novel recombinant viral vaccine to be developed completely from scratch in India. And we have a further improved variant of the formulation, which which contains some of the conserved regions that I talked about. And these will be tested. We propose to test them in the EU and in India with support from DBT. Hopefully we actually get the money from DBT. And so just to summarize, there have been really spectacular advances using AI and machine learning in structure prediction and in a variety of other areas in function prediction. Despite this, so far it hasn't been particularly successful at predicting single point mutations which are stable. And so also, though there have been a lot of transformational advances that have in viral vaccine development, including the development of mRNA formulations, which I haven't talked about, there's still very important unsolved problems that remain. For example, finding an efficacious vaccine for HIV-1. And very often the rate-limiting step is the discovery research, which allows us to find the right molecule to put into the vaccine that will elicit a protective immune response. And all the other steps, they can be done by industry. And even when we think we know everything about the molecule, so for example, the RSV vaccine that was developed by GSK, Pfizer, and Moderna, it worked very well in adults, including the elderly. But when they did the trial in infants, they found that the very same vaccine actually didn't work, and in fact, there were some hints that it might have caused enhanced disease in this population, despite all the advancements in, vaccine design and experience that had been accumulated over the past many decades. And I think they're still trying to figure out what exactly went wrong. So, despite all these advances, there's a lot that we need to know, and there's no substitute really for a clinical trial. So India has very substantial strengths in manufacturing. We saw that during the COVID pandemic, but there's very little discovery research in industry because it's much more efficient for them to take molecules that have been clinically tested elsewhere in the world and then just scale them up and use them. And so we need to have better collaboration, better funding, and there needs to be really one of the difficulties we had in advancing our vaccine formulation during the pandemic was the regulatory hurdles that we encountered, amongst many others. But gradually those things are improving, and I hope I've been able to convince you that while it's not easy, with very limited funding, we can develop, manufacture, and clinically test novel viral vaccines in India. So I'd like to thank, of course, all the folks who did the work. These are the people from my lab at IISc and some of my collaborators at IISc, a variety of collaborators within India from different institutions, the Vaccine Formulation Institute in Switzerland, through we access the adjuvant for our vaccine, and the Gates Foundation for some of our COVID work. Collaborators at NCBS, INSTEM, NII, and of course, none of this would have been possible without the very sustained effort at Minvax, which is a startup that I co-founded, which has been, really key our ability to advance this vaccine into clinical development. So that's it, and so this is we got worldwide media coverage for our COVID vaccine, but, no money, and we couldn't do anything with it. So it's a cautionary lesson for everybody. But it's been fun. And thank you for the opportunity to present before all of you, and I'll be happy to take questions.
Very enriching, very thought-provoking, very enticing. I would like to congratulate you for that. A couple of very simple questions. When we talk about a vaccine which works in the adult, it does not work for children. Would you not consider the possibility of gut microbiome, because the gut microbiome in children is very different than adults? Then we have circadian, rhythm. Children sleep for longer, adults probably less than 8 hours. And third things which have gained momentum is the presence of microplastics in the human body. So inside our body, a cell has to probably witness and tolerate all kind of molecules. And last question, your talk provoked me to clarify my doubt. I plead my ignorance. When we have a simple eukaryotic cell, it has close to about 40 million protein molecules. And these molecules are contributed by close to about 6,000 genes. Now the question is, how the compartmentalization of these protein molecules are maintained. I would love to hear from you because already you have charged the entire atmosphere. I posed
Sure.
4, 5, 6.
So your first question, obviously we don't know the answer to that. All the points you raised are totally valid. I think one other very important point, because of the regulatory hurdles, you would think that in Australia the regulatory standards would be so high that it would be much more difficult to do a trial. But in fact, in India we have some fundamentally unreasonable requirements because the regulatory pathways for biopharmaceuticals in India have been tailored to generics. So where you know in advance that the molecule is going to work, and so there are a lot of hurdles that are inappropriate for a new molecule that are in place. And so therefore it was, by the time we got permission to do a safety toxicity study in mice in India, which took us 2 years. That was partly because we were new to the area. By that time, we had completed our phase 1 trial in Australia. So the situation is certainly improving, and I hope in the future that we don't have to do this, but that's...
So very wonderful. Thank you very much.
Sure.
Couple of questions. One is that what do you think, there are news about mRNA vaccines which has side effects quite a bit.
Right.
What is your comments on that? And the second thing is, what do you think about the future of nanobodies and the recent trends of single-chain antibodies in terms of vaccine potential?
Right. So, every molecule that you put into your body is going to have some side effect in some number of people. Now, in the case of mRNA vaccines, I think one of the things is that if they are improperly administered, the mRNA can go to all parts of the body. And so, in a protein vaccine, it doesn't, the immune response is, you inject it and then it doesn't, go all over the place. So, because the mRNA vaccine is producing an antigen in situ, if the mRNA is transported to some other part of the body, you can maybe get some undesirable responses. But it's very hard to, to answer the question properly, because vaccines are, especially the mRNA vaccines, were administered during a pandemic. And in the pandemic, it's impossible to figure out the effect of infection and vaccination, because everybody who has been immunized is also exposed to the virus. So the result that you get, whether it's due to a vaccination or whether it's due to, a subcritical, is not very easy to figure out. So I think, I think one thing is certainly true is that the reactogenicity of mRNA vaccines is much higher. So people do get, knocked out for a few days, and that's a definite downside. Whether there is actually a significant safety issue with that, I think that's been, a little more difficult to establish. Not really, not clear about that aspect. I think the big advantage with mRNA vaccines, as somebody who's going through this process and clinical development, the big advantage with mRNA vaccines is all mRNA is the same. And because the protein is produced in the body, the manufacturer is not subjected to the burden of characterizing the protein. All you have is the mRNA, and if you characterize one mRNA, you know how to characterize any other mRNA. With a protein, the problem is every protein is different, and so the process development takes a much longer time. It's not that was why the mRNA vaccines could be put into people so much more quickly than the protein vaccines.
The second thing is about nanobodies.
About nanobodies, yes. So nanobodies are, they're certainly very good at, you can get very good binders, and you can, because they're smaller, they can bind to regions of the protein or target that are not accessible to antibodies. The problem with nanobodies in a therapeutic, it's a problem and sometimes an advantage, is that they get cleared by the body very rapidly. So if you want persistence of the therapeutic, then nanobodies are not a good way to go. If you want the therapeutic to be, then it's certainly something to be considered. They're also, because nanobodies are not what we naturally make, there's the problem of immunogenicity happening from repeated administration of the nanobody, which is less likely with the antibody, which is a human molecule made by the body.
But the concept which people are thinking is to put active immunization along with nanobodies for immediate control of pandemic.
Right. But with that, the problem is that there needs to be some persistence, because you don't know when you're going to be infected, right? Even in a pandemic. It may be today, it may be 3 weeks from now, 2 months from now. So I think unless the persistence of the nanobodies in the body is solved, that becomes difficult. That is not a problem with antibodies.
Okay, so I think I'll take this opportunity to thank Raghavan for a very, very interesting talk.